Good morning everyone. Today we are going to study about cell-mediated immunity. Cell-mediated immunity is mediated through activated T lymphocytes. So from the last video we understood that T lymphocytes are matured in thymus. So after their final maturation, these mature T lymphocytes are released into the blood, and through the blood circulation they reaches the lymphoid tissues like lymph nodes, spleen, etc. So once these T lymphocytes get exposed to the antigen that is specific to it, it will bind to the antigen. So once it binds to the antigen, it gets activated. So cell-mediated immunity is mediated through such activated T lymphocytes. There are basically 4 types of T lymphocytes in your body, that is helper T cell, cytotoxic T cell, suppressor T cell, and memory T cell. So helper T cells form 3/4 of the T lymphocytes. So the majority of T lymphocytes are helper T cell. They are the major regulator of all immune functions. As the name indicates, they helps in the immune function by releasing proteins known as lymphokines or cytokines that helps in cell communication or cell signaling. So these cytokines released from these helper T cells will act on other cells of the immune system. This helper T cell is again classified into 2 subtypes, that is Th1 and Th2 cell. Th1 cell secrete cytokine like interleukin 2 and interferon gamma, and they are helping in cellular immunity or cell mediated immunity. Th2 cell secrete interleukins like interleukin 4, 5 and 6, and they help in humoral immunity. Most of the helper T cells display the glycoprotein marker CD4, so they are called CD4 T cell. So helper T cells are also known as CD4 T cells because of the presence of CD4 glycoprotein marker on its surface. Moving on to cytotoxic T cell. Cytotoxic T cell is a direct attack cell that is capable of killing microorganisms and even some of the body's own cells. So they are known as killer cells. So they can either destroy microorganisms or body's own cells, that is infected cells. Receptor protein will be present on the surface of the cytotoxic T cell, which will bind to the appropriate binding specific antigen on the target. After binding, they will secrete perforins. Perforins are hole forming proteins. So these perforins will create holes in the membranes of the target cell. So through these holes, fluid flows into the cell from the interstitial space, and the cell will become swollen and it dissolves. Simultaneously, it also releases cytotoxic and digestive enzymes into the attacked cell or target cell, which will again destroy it. So these are the 2 mechanisms by which cytotoxic T cell destroy the target cell. So target cell can be a microorganism or a infected cell. So they kill virus infected cells, cancer cells and transplant cells or other types of foreign cells. Development of this cytotoxic T cell is helped by helper T cell by releasing interleukin 2. So we know that interleukin 2 is released by Th1 subtype of helper T cells. So Th1 subtype of helper T cell is required in activation or development of cytotoxic T cell with the help of interleukin 2 released by them. And these cytotoxic T cell display the glycoprotein marker CD8, so they are also known as CD8 T cell. Now in this diagram you can see this bigger cell is the target cell or attacked cell. You can see these are the cytotoxic T cell, and on the cytotoxic T cell you can see receptor, okay, and here on the target cell you can see the antigen. On the cell membrane you can see pink dots, they are the binding specific appropriate antigen on the target cell. So the receptor on the cytotoxic T cell will bind to antigen on the target cell. Now after their binding, they will release perforins into the target cell, which will create holes in the cell membrane of the target cell. So water enters from the interstitial space, this target cell swollen and it will dissolve. Also they will release cytotoxic and digestive enzymes into the target cell, which will again destroy them. So this is the mechanism by which cytotoxic T cells, once get activated, they will destroy the pathogen. Moving on to suppressor T cell. As the name indicates, they suppress the functions of cytotoxic T cells and helper T cell. They prevent the cytotoxic T cell from causing excessive immune reaction against body's own tissues. For this reason, suppressor T cells along with the helper T cells are called regulatory T cell. So they help in immune tolerance, that is limiting the ability of the immune system to attack body's own cell. So that is known as immune tolerance. Moving on to memory T cell. So after exposure to a given antigen, they get activated. So when they are exposed to a given antigen, these T lymphocytes get activated, and a small number of activated T cells persist as memory T cells. After activation, actually they go into the next stage, that is proliferation, under the influence of a cytokine known as interleukin 2. So here, when they bind to the antigen, they get activated, and before moving on to the proliferation stage, some of the activated T cells, they persist as memory T cell. These memory T cells are converted to effector cells when the same antigen enters the body for the second time. So the effector cells are nothing but these helper T cells only, and then the rest of the function is same. These memory T cells are present in areas where invading organisms enter the body, like mucosa of GIT and respiratory tract. Long life of memory cells is due to their repeated exposure to small amount of antigen. Moving on to mechanism of cell-mediated immunity. We discussed mechanism of cell-mediated immunity under 3 headings, that is antigen presentation, antigen recognition, activation and attack. So what is antigen presentation? T lymphocytes, they respond to an antigen only when they are attached to a protein molecule called MHC protein. So MHC means major histocompatibility complex. So MHC protein. We know that T lymphocytes will be present in the lymphoid tissue like lymph node waiting for the antigen. So they respond to antigen only when they are attached to a protein known as MHC protein, which will be present on the antigen presenting cell. That means the T lymphocytes will not respond to a free antigen. So this antigen presenting cells include dendritic cells in the lymph nodes and spleen. They are the most potent antigen presenting cell. Langerhans cells in the skin, macrophages and B lymphocytes. Now what are these MHC protein? So MHC proteins are classified into class 1 and class 2. Class 1 MHC protein will be present on all the nucleated cells and platelets. So all your nucleated cells will be having MHC1 protein on its surface, and they are attached to peptide fragments which is generated from those proteins that are synthesized within the cells. For example, peptides from mutant or viral protein. MHC2 protein is present only on the immunologically active cells like activated T lymphocytes, B lymphocytes, macrophages and other antigen presenting cell. And MHC2 proteins are attached to the peptide fragments of extracellular antigens like bacteria, and they are digested in the late endosome. So when an antigen first enters the body, it is taken up by antigen presenting cell, and these antigens are partially digested inside this antigen presenting cell. And these peptide fragments will be attached to the MHC protein, and that complex will be transported into its surface. Now the antigen presenting cell will be having an MHC protein on its surface which is attached to an antigen, and this antigen presenting cell will carry this antigen and present the antigen to the helper T cell that is present in the lymphoid tissue, and these helper T cells will bind to the antigen, and once they bind to the antigen, they get activated. So the presenting of this antigen to the helper T cells by attaching it to an MHC protein, that process is known as antigen presentation, and that is done by antigen presenting cell. So you can see the diagram. Here the antigen is taken into the antigen presenting cell, and you know there is lysosome present in the cell which contain digestive enzyme, and with the help of phagocytosis the antigen is taken into the antigen presenting cell, for example macrophages, and this is phagosome. Phagosome is a vesicle which contain the antigen in it, and there will be fusion of phagosome with the already present lysosome, and we get phagolysosome, and they will release, this lysosome release its contents into the bacteria, and this bacteria is broken down into small fragments. Okay, and these fragments are attached to MHC protein, and that complex is brought to the surface of the antigen presenting cell, and the leftover fragments are released by exocytosis. So this is how the antigen is taken into the cell, that is antigen presenting cell like macrophage, and finally we get the MHC protein and antigen complex on the surface of the antigen presenting cell, which is presented to the helper T cell present in the lymphoid tissue. Again you can see this is an antigen presenting cell. This is a microbe taken into the antigen presenting cell. It is broken down into pieces with the help of lysosome present in the antigen presenting cell. This is the MHC protein. This is a peptide fragment of antigen. Both are attached, and that complex is brought to the surface. So this is a class 2 MHC molecule on the antigen presenting cell. This is the antigen, and this complex is coming towards the helper T cell. Helper T cell will be having a receptor specific to that antigen, and this receptor binds to this complex, and helper T cell gets activated. Next is antigen recognition. That is, if the antigen is presented to cytotoxic T cell, then that antigen should be attached to MHC1 protein, which will be present on all the nucleated cells and platelets. And if the antigen should be presented to helper T cell, then that will be attached to MHC2 protein, which will be present in all the immunologically active cell. So only when this binding is taken place and it is presented to the cell, they respond. This binding of antigen and T lymphocytes is called antigen recognitions. So once they are presented to the helper T cells, they will bind to the antigen because they have corresponding receptor on its surface which is specific to that antigen. So that receptor will bind to the antigen MHC complex. So once they bind to that antigen MHC complex, these helper T cells will become activated helper T cells. So for recognition, they should be attached to the MHC protein. If it is cytotoxic T cell, MHC protein should be MHC protein 1. If it is helper T cell, then MHC protein should be MHC protein 2. So that is antigen recognition. Moving on to the final stage, that is activation and attack. So two signals are necessary to produce T cell activation. One is produced by binding of the antigen to the T cell receptor, that we have already said. The other one is produced by joining of the surrounding proteins called adhesion proteins in the synapse. Synapse means the junction where the helper T cell is binding to the antigen which is present on the antigen presenting cell attached to the MHC protein. So that is a synapse. So there will be adhesion proteins on the surface of helper T cells and also on the surface of antigen presenting cell. So they also will bind. So these two binding is necessary for the activation of helper T cell. If the first signal happens but second does not, means T cell is inactivated and become unresponsive. So there should be binding with the antigen and receptor on the helper T cell. Also there should be binding with the adhesion proteins which are present on both helper T cell and antigen presenting cell. So two binding should happen for the activation of helper T cell. So you can see this is the antigen presenting cell and this is the T cell. Okay, so this is the MHC protein. MHC protein is having antigen attached to it. Here you can see receptor on the T cell. So this receptor will bind to the antigen which is attached to the MHC protein. So this is one binding. So here they have shown cytotoxic T cell. So what type of MHC protein will be this? This will be MHC1. If it was helper T cell, then we should expect MHC type 2 on the antigen presenting cell. Then you can also see the second binding, that is the binding between adhesion proteins on both cells. So these two bindings should happen for the activation of the T cell, whether it is helper T cell or cytotoxic T cell. On exposure to antigen, they will bind to the antigen, then these T cell will get activated. Now the T lymphocyte of a particular clone, they proliferate and release large number of activated T cells into the lymph. So clone means a group of T lymphocytes that is specific to a particular antigen. So that is a clone. So after binding, they get activated. After activated, they will release a cytokine that will again helps in the proliferation of helper T cell. And after proliferation, there will be a lot of helper T cells in the lymph. Then this activated T cells then pass into the circulation, and they are distributed throughout the body, passing through capillary walls into the tissue spaces, back into lymph and blood. So here you can see a diagram where they are showing all the three stages, that is antigen presentation, antigen recognition, activation and attack. So here you can see macrophage. So macrophage is the antigen presenting cell here. Now this antigen presenting cell will be having a MHC protein. So here they have shown MHC class 2. So if the MHC protein type is class 2, means the T cell will be helper T cells. So we can see here, yes, since CD4 is the glycoprotein marker they have shown, this is a helper T cell. Now this antigen will be taken up by the antigen presenting cell with the help of lysosomal enzyme. That antigen will be broken down into multiple pieces, and it will be attached to MHC type 2 protein, and that complex will be taken into the surface of the antigen presenting cell, that is macrophage. You can see this macrophage is having MHC2 protein with antigen attached to it. Now they will present the antigen to this helper T cell. So helper T cell will be having a receptor that is specific to that particular antigen. So this receptor present on the helper T cell will bind to the antigen. So during this binding, that results in activation of this helper T cell. And simultaneously you can also see interleukin 1 is released by the macrophage, which is going and acting on the helper T cell. So both will result in the activation of helper T cell. So you can see they have labeled here activated T cell. Now once they are activated, they will release an interleukin known as interleukin 2. Okay, so the receptor for interleukin 2 is present on two sides. So one is on the surface of this same activated T cell, and also it is present on the surface of cytotoxic T cell. So the interleukin 2 released by this activated T cell will go and act on the same activated T cell. Also they will go and act on the receptor that is present on the cytotoxic T cell, and that will helps in the development of cytotoxic T cell. So we have already studied that interleukin 2 helps in the development of cytotoxic T cell, and we know that interleukin 2 is released by Th1 cell. And you can also see here an antigen presenting cell having MHC class 1 protein on the surface is presenting the antigen to cytotoxic T cell. So they have labeled CD8. So this is a cytotoxic T cell. So the MHC protein should be MHC1. So MHC1 is there on this antigen presenting cell, and we know that MHC1 is present on all the nucleated cell and platelets. So this can be any nucleated cell which is infected. So they are presenting that infectious agent, that is the antigen, to this cytotoxic T cell. So cytotoxic T cell will be also having receptor specific to that antigen. So they will bind to this antigen, and that results in the activation of the cytotoxic T cell. Once cytotoxic T cell is activated, they will release perforins that will make holes in the membranes of the cell, leading to lysis of the cell, and also they will release cytotoxic and digestive enzymes. So that is a mechanism by which cytotoxic T cell destroy the antigen, and this is how cytotoxic T cell is getting activated under the influence of interleukin 2 released by the activated T cell, and also antigen presenting cell presenting the antigen to the cytotoxic T cell after binding with the MHC1 protein, and this antigen presenting cell can be any nucleated cell. Before proceeding further, we have to understand that when exposed to an antigen or an organism, our immune system will select the effector mechanism that is appropriate for that particular pathogen. Maybe via activation of cytotoxic T cell and will destroy the pathogen by two mechanisms, or by activation of macrophages that will cause phagocytosis of the organism, or via the antibodies. So it will select which mechanism is appropriate for that particular antigen. This selection is important because activation of an inappropriate mechanism for that pathogen can lead to increased susceptibility rather than protection from the pathogen. So this selection is very important. Depending upon the effector mechanism that is selected for destruction, body will decide which subset of helper T cells should be proliferated. That is, we know that helper T cell is again divided into Th1 subset and Th2 subset. So Th1 will release interleukin 2 and interferon gamma. Interleukin 2 will help in the activation of cytotoxic T cell and it will destroy the pathogen. Interferon gamma will activate macrophages and it will cause phagocytosis of the pathogen. And Th2 cells secrete interleukin 4, 5, 6 that will activate B lymphocytes, which will proliferate to become plasma cells and memory B cells. Plasma cells will release antibodies, and these antibodies will cause destruction of the pathogen either directly or indirectly. So depending upon the effector mechanism, immune system will decide which type of helper T cell, that is Th1 or Th2, should be proliferated. Pattern of cytokine triggering by pathogen and local concentration of steroid and vitamin D3 metabolites in the lymphoid tissue will decide which type of helper T cell or which subset of helper T cells should be proliferated. If an organism triggers the release of interleukin 2 and interferon gamma, then helper T cell 1, that is Th1 cell, is proliferated. If an organism triggers the release of interleukin 4, 5 and 6, then Th2 subset will be proliferated. So everything depends upon the effector mechanism that is selected for destruction of that particular pathogen. So here you know that after binding, this T cell is becoming activated. Now this activated T cell will release interleukin 2. Interleukin 2 will act on the same activated T cell. So once this happens, this T cell will get proliferated, and which subset is decided already by the immune system, either Th1 or Th2. If it is via macrophages or cytotoxic T cell, then Th1 cells will be proliferated. If the destruction is via antibodies, then Th2 subset will be proliferated. And you can also see this IL-2 will be acting on the cytotoxic T cell and they will get activated. So this is how the type of helper T cell is selected for proliferation. So if it was Th1 cell, then they will destroy the pathogen either via activation of macrophages, because macrophage is activated by interferon gamma, which is released by Th1 cell, or they will cause a destruction with the help of cytotoxic T cell, because cytotoxic T cell is activated by interleukin 2, which is again released by Th1 cell. If it was Th2 cell, then they will get proliferated and they will release interleukin 4, 5 and 6, and they will go and activate B cell. So B cell get differentiated to plasma cell and memory B cell, and this plasma cell will release antibodies and they will destroy the pathogen directly or indirectly. So depending upon the effector mechanism that is selected, they will decide which subset of helper T cell should be proliferated so that they can destroy the pathogen with that selected mechanism. To summarize, cytotoxic T cells can be activated by forming a synapse with MHC1 protein. They kill the target by forming perforins and by releasing digestive enzymes. Helper T cells can be activated by forming a synapse with MHC2 protein. They secrete cytokines and helps in both cell-mediated and humoral immunity. So helper T cells are divided into Th1 and Th2. Th1 will release interleukin-2 that will activate cytotoxic T cell. Then they will release interferon gamma that will activate macrophages leading to phagocytosis. These two cells will release interleukin 4, 5 and 6 that will activate B cells, causing its proliferation to plasma cells and memory B cells. Plasma cells will release antibodies, and these antibodies will cause destruction of the pathogen either directly or indirectly. So whenever a pathogen enters your body, your innate immune system will try to destroy the pathogen. So when that fails, your acquired immune system comes into action. Pattern of cytokine triggering by pathogen will select which effector mechanism to be used for destruction of the pathogen. It can be either by activating cytotoxic T cell or activating macrophage or by activating B cell. So depending upon that effector mechanism, the immune system will decide which subset of helper T cell should be proliferated after activation of T lymphocytes and release of the interleukin-2. Because once interleukin-2 acts on the helper T cells again, that will result in the proliferation. But which subset should be proliferated, that will be decided by your immune system depending upon the mechanism that is selected for the destruction. So that is very important in cell-mediated immunity. When you get a question like write a short note on cell-mediated immunity, you have to start with the definition of immunity. Then you have to write a classification. That is, it is divided into innate and acquired. Acquired is further classified into cell-mediated and humoral. Then you have to write about cell-mediated immunity. You write a short introduction. That is, it is mediated by activated T lymphocytes and a few points about all 4 types of lymphocytes. Then you write about the mechanism of cell-mediated immunity in 3 headings. That is antigen presentation, antigen recognition, activation and attack. You have to draw a neat labeled diagram. You can draw the diagram that is showing the 3 mechanisms, and you can also draw diagram showing binding of helper T cell and antigen presenting cell. You should also mention about the selection of the effector mechanism. I hope it is clear. Thank you.